Welcome to Virology Laboratory. I'm Mustafiz. I'm the head of Virology Laboratory, and we have a very young and energetic team for doing this virus research. So we have mainly three pillars. The one is molecular virology, and in this group, there are separate teams like enteric virology who are working with rotavirus, norovirus, adenovirus, which are causing diarrheal disease mainly. And another team is working with respiratory viruses. They're dealing with influenza, RSV, and other respiratory viruses like COVID-19, and they are detecting these viruses and also characterizing these viruses using sequencing. And the third team, like sexually transmitted disease, they are working and dealing with the virus like human papilloma virus, and then HIV, and other bloodborne pathogens like dengue, chikungunya. So all these viruses that are important, they are dealing with these. And another pillar is the vaccine evaluation team. That means they have developed some assays, installed these assays, which are used to evaluate the vaccine trial samples. The third group is engaged in detecting, or this is called etiology, the viral and pathogen etiology team. They're also working with different surveillance like child mortality surveillance they're dealing with. So these teams are working together to give a huge input in the public health domain in Bangladesh. So I hope you will go to that group to know more about this group. And thank you very much.
Welcome to the Molecular Virology, where scientific innovation meets global health. The day begins with a team meeting led by the supervisor to review ongoing projects, align priorities, and define a structured workflow that ensures seamless and efficient lab operation. The specimen processing room is a BSL2 laboratory. The workflow starts with sample receiving. Once we get the samples, you're immediately moved into the biosafety cabinet. Inside the cabinet, we check that the body tag matches a log sheet serial number to ensure correct identification. After verification, we move on to the processing steps like vortexing and centrifugation. Then, we aliquot the samples, getting them ready for extraction. We use the Chemagic 360 system for automated extraction, which isolates nucleic acid with high accuracy and efficiency. The extracted nucleic acid is then used for specific virus detection using real-time PCR. To move forward with the real-time PCR, we first need to prepare a master mix. This work takes place in a master mix room, a dedicated controlled space designed specifically for reagent handling. Once ready, the master mix is transcribed to the PCR plate for dispensing, completing the setup for molecular testing. Then we add extracted RNA in the PCR plate. This is done in the clean and controlled template herd. Once the template is added, the PCR plates are placed into the real-time PCR machine to begin the testing process. After the run is complete, the results are carefully analyzed by our expert team to ensure accuracy and reliability. In addition to our project-based work, we routinely perform conventional PCR using a dedicated facility specifically designed for this purpose. Following amplification, we proceed with agarose gel electrophoresis for product identification. The gel is run in a standard gel tank. Once the run is complete, we visualize the bands using the BioRad gel doc imaging system. For precise analysis of the PCR bands, we utilize specialized software tailored for gel documentation and interpretation. Following PCR band confirmation, selected samples are prepared for either whole genome or Sanger sequencing using our sequencing facility. We have two applied biosystem sequencing machines that support this process. This facility is utilized not only for our own research projects, but also offers commercial sequencing services to external researchers and collaborators. In whole genome sequencing, once the samples are assessed and the library is prepared, the sample library is gently loaded onto a flow cell with utmost precision, which is inserted into an Oxford Min-ion nanopore device. After completion of sequencing, the raw sequence data is computationally assembled into complete genomes and then used for identification of global lineage nomenclature. High-quality consensus sequences are submitted to international public databases such as GIS-8. This submission of the sequences data supports the global genomic surveillance network, enabling real-time monitoring of pathogen evolution, spread, and identification of variants of concern. Further analysis, including phylogenetics, help clarify evolutionary links and guide interventions like vaccine strain selection.